Okay, so now in this lecture, we will talk primarily about tRNAs and ribosomes. Okay, before we move on to actual process of translation in the third lecture onwards. Okay. So, what is the objective of translation or protein synthesis? That is to align the amino acids along the mRNA and form peptide bonds between them. Okay, so if it were entirely to happen by the diffusion and chance polymerization, it would take a whole lot of time, right? So there is therefore a mechanism to align the amino acids along the mRNA, which is the code, the coding strand or the sense RNA or whatever you call it. And that alignment is done by another kind of RNA called the tRNA or the transfer RNA. Transfer RNA is the adapter molecule. It is the real translator between the RNA, that is the code, and the amino acid that is to be polymerized. So tRNA actually speaks both the languages. Okay, it speaks the language of the RNA because it forms, like hybridizes with the mRNA based on sequence, and it carries an amino acid which is to be polymerized. And that actual process of polymerization is done by an RNA which is present in a ribonucleoprotein complex called the ribosome. Okay, I'm pretty sure you know that the actual peptidyl transferase activity or the amino acid polymerization activity resides within the RNA of the ribosome. So translation or protein synthesis or polymerization of amino acids based on an RNA template is dictated by three different RNAs. One is the messenger RNA that provides the code. Second is the tRNA that aligns the amino acids as monomers along the code, and ribosome that actually causes the polymerization between the individual amino acids. Okay, so what happens that the tRNA, it brings one amino acid to the ribosome, aligns itself to a particular 3-nucleotide sequence on the mRNA, and then the next tRNA comes and aligns itself to the next codon, and then there is a polymerization reaction happens from two different monomers. Now you have a bipeptide polymer, and then it moves. Then the next amino acid bearing tRNA comes, and so on. It keeps on happening in a processive manner. Okay, so in this schematic, you can see that all the different RNAs, how they are playing their role. mRNA, as I told you, is the template for this particular purpose, for the protein synthesis purpose. tRNA that comes with their own anticodon loop that aligns and hybridizes with the codon, but it also carries the amino acid, and that is added to the elongating polypeptide chain, and then the polymerization proceeds, and it keeps on happening till the ribosome reaches what is the stop codon, and there there is no tRNA corresponding to the stop codon, but release factors are there which releases the peptide from the elongating polypeptide and from the final tRNA, and your protein is synthesized. Okay, so the tRNA is called the adapter molecule, as I told you earlier, and it has a very intricate three-dimensional structure, and that is critically important for its function. The anticodon loop conventionally is written in the 3' to 5' orientation, so that the mRNA can be shown 5' to 3', and the base pairing can be shown, but then it is in the 3' to 5' orientation is how the tRNA anticodon loop is shown. Okay. The tRNA, if you remember, Robert Hawley got his Nobel Prize because of deciphering the tRNA structure and all the different chemical modifications it has. It has many modified nucleotides. Like it has the uridine is modified to dihydrouridine. and it is present in a particular location within the tRNA, which is called the D loop. And then there is pseudouridine and ribothymin that is present in the T-CG loop. And all these are modified bases. And of course, there is the anticodon loop. And please note presence of inosine, which is not a nucleotide present in other RNAs, but in tRNAs, which is deaminated adenine. So inosine, the importance of that will become clear in the next few slides. So these are the different, like this is the three-dimensional structure and the different chemical modifications that are there in the tRNA. Okay, and this the amino acid is accepted at this adenine location, and there is this CCA, which is called the CCA terminus. The CCA is often added as a post-transcriptional modification of tRNA. So the tRNA is actually transcribed from its DNA template, and after the tRNA is made, the CCA terminus is added to it, which acts as the acceptor location for the amino acid. So of course, you do not need to remember any of these structures, but just you should know that there are different kinds of modified amino acids, and the tRNA, mRNA, like where there is this modified nucleotides, no Watson-Crick base pairing happens there, and like tRNA has this clover leaf-like structure. Okay. Now comes to the importance of inosine. Okay, so now we know that there are 61 different codons. So if all the 61 codons are to be optimally utilized, then ideally you should have 61 different tRNAs in any cell. But in reality, much fewer than 61 different tRNAs are present. So then how is this, apparent, lipoplexing situation resolved? So what you get to see, please note in the tRNA, okay? In the tRNA, in the 3' to 5' location, as I said, the tRNA is typically shown in 3' to 5' direction. So from the 5', the first nucleotide in the tRNA marked in red, and in both these cases, just make a note of this. Now, it has been observed that if this first nucleotide is a C, then in the RNA, the codon will typically have a G. If the first nucleotide is an A, then in the RNA codon, the third nucleotide will always be a U. If the first nucleotide is a G, then the third nucleotide in the RNA, in the codon, can be either a C or an U. If the first nucleotide in the tRNA is an U, then the third nucleotide in the codon can be either a A or a G. But if this first nucleotide is an I, then the third nucleotide on the RNA codon can be either of a C or a U or a A. Okay, and similarly, if you put it the other way around, then if the third nucleotide of the mRNA is a C, then this first nucleotide in the tRNA can be either a G or an I. So G is the normal cognate, but I is, it fits in with so many things, so promiscuous. Okay, if it is an A, it can be a U, which is the cognate one, and an I. So on and so forth. So therefore, there is a non-Watson-Crick base pairing happens in the third position of the codon, and which is the first position of the tRNA, which is called the Wobble position, and there is Wobble base pairing, and there you do not have adenine, rather what you have is a deaminated product of adenine called inosine, and that's what gives the flexibility of a particular tRNA, a particular molecule of tRNA to base pair with several kinds of synonymous codons. So, for example, as you know that leucine has 6 different codons, okay? But, like the uh, there are 4 codons where the third position is either an A or a C or an U. In either, in any of these cases, the tRNA in its first position always has an I. Okay, so just having one tRNA for leucine, having the anticodon loop of IgAG can base pair with 4 different codons. Okay, so this is how, even though the number of tRNAs are much less than 61, it can satisfy all the different, it can decode all the different 61 codons that are required for polymerization of the entire repertoire of amino acids. Okay. Now comes the coupling of the tRNA with the amino acid. There is an enzyme called amino acyl tRNA synthetase that couples the tRNA with the amino acid. And this is a very, very important reaction. Why? Because see, the tRNA, of course, will recognize the codon based on, complementary sequences. But if this coupling is wrong, then the tRNA can potentially bring a different amino acid to the growing polypeptide. Therefore, the fidelity of translation is just as critically dependent on amino acyl tRNA synthetase for it to couple the right base to the right tRNA. If in this step, if there is a mistake, like AAA, instead of coupling to a phenylalanine amino acid, if it couples to say serine, then all bets are off. Then you will be incorporating wrong amino acids. In fact, experiments have been done that after a serine is, or not serine, cysteine is coupled with its cognate tRNA, and if it is oxidized after coupling to, like say, cysteine is oxidized to serine, you understand that it's just a sulfidyl group versus a hydroxyl group. Then in the growing polypeptide, you can actually incorporate serine instead of cysteine. Therefore, once this coupling is made, then whatever amino acid is coupled to the tRNA, that's what will be incorporated in the growing polypeptide chain. Therefore, this coupling reaction catalyzed by amino acyl tRNA synthetase is very, very critical. And I will come to some of the nuances of it in a couple of slides. So how does this reaction happen? So first, amino acyl tRNA synthetase activates the amino acid by coupling it with AMP. Okay, and pyrophosphate is released in the process that drives the reaction. So now you have amino acyl tRNA synthetase, which is temporarily covalently linked with amino acid, which is and AMP, which is covalently bonded with AMP. And then comes the tRNA with its CCA terminal, and then on the CCA terminal, the amino acid can be added either to this 3' position hydroxyl or the 2' position hydroxyl. Depending on where the amino acid is added, it is called a class 1 synthetase or a class 2 synthetase. It is not really that, for our purpose, it is not really that critically important. But you should know that there are 2 kinds of amino acyl tRNA synthetases, and that couples the amino acid to the tRNA, and amino acyl tRNA synthesis is recycled and it goes back to doing its job again. Okay, so in terms of order of addition, how it happens is that in the amino acyl tRNA synthetase first binds to the amino acid and ATP, and then it gets activated. Upon binding to the cognate amino acid, then only the pocket opens up for it to bind to the cognate tRNA. And then the tRNA and amino acid is coupled by the amino acyl tRNA synthetase to make the charged tRNA, which is then released. So therefore, this is a a classical example of your induced fit model, that only when the amino acid, the correct amino acid is bound to the correct amino acid tRNA synthetase, only then the correct tRNA can come and bind to the tRNA binding pocket for the actual synthesis to happen. Now you think about it, that there are only 20 amino acid tRNA synthetases. Okay, there are only 20 amino acid tRNA synthetases, but because of degeneracy of the codon, so multiple different tRNAs can bind to the same amino acid, can be charged by the same amino acid. Like the number of tRNAs are not 20. Okay, number of tRNAs are more than 20, perhaps less than 60, of course less than 61, but more than 20. So, and you think of the particular example, that this one phenylalanine tRNA will be able to bind 4 different codons, and in all the cases it will be charged with phenylalanine. But there are 2 more codons. Okay, they will also be charged by phenylalanine only, and the enzyme to charge it is amino acid tRNA synthetase. So how the synonymous tRNAs are charged by the same amino acid, meaning there is only one amino acid tRNA synthetase for phenylalanine, and it will have to potentially bind to at least 2 different tRNAs and still be charged by leucine only, phenylalanine only. So how does this happen? So that's where something called the identity elements come in picture. So the close inspection of the sequence of tRNA has shown that tRNAs that are charged by the same amino acid, they have very high sequence conservation in certain areas that are shown in these large yellow balls, and that is what is referred to as identity elements. Anticodon loop is certainly important, but there are other areas of the tRNA which are also important, and they are referred to as identity elements. Okay. And then, of course, the ribosome, which actually does the polymerization reaction, and it is not RNA alone; it's a RNA protein complex, and it has in bacteria, you have whether it is prokaryote or eukaryote, ribosomes are in 2 subunits: a small subunit and a large subunit. In bacteria, the large subunit is 50S, small subunit is 30S. In eukaryotes, the large subunit is 60S, small subunit is 40S. This S refers to Sedgwick constant, which is basically a centrifugation term, meaning how slow or fast it precipitates in a viscous liquid in a centrifuge. Okay. So in bacteria, the large subunit has 31 different proteins and 23S RNA and 5S RNA. Okay. And because it is part of the large subunit, so they are called L1, L2, like L31. The small subunit has 21 amino acids, L1, L2 up to L21. And then it has 16S ribosomal RNA. So these ribosomal subunits can either exist as independent subunits or can exist as a complex like ATS ribosomal subunit. And in bacteria, that you will see later, there is an active mechanism to keep them separated. In eukaryotes, so far, that kind of a mechanism is not known. Okay. The way it works is that on the small ribosomal subunit, there is a decoding site. That's where the mRNA, which is kind of going around the 30th subunit, and the tRNA, they come together, and then the peptidyl transferase location is shown here, and the nascent polypeptide grows inside the ribosome. There is an exit channel, and through which the growing polypeptide will eventually come out. So in the ribosome, and please note very carefully that these features are only in the context of when the 2 subunits are joined together, like in 70S in bacteria or 80S in eukaryotes. there are three sites. There is an A site, a P site, and an E site. A site, it is very simple to remember. A site is the amino acyl tRNA binding site. So when the tRNA is coming with a monomer, then that enters the A site. P site is the peptidyl tRNA binding site. So therefore, it is there the tRNA has a polymer attached to it. And the E site is where the tRNA is ejected from the ribosome. So the exit site. Okay. So in this context only, it is important to remember that the first tRNA, if it comes to the A site, then there is no way that the next tRNA can come and form a dipeptide with methionine. Therefore, in the first tRNA, of course, it is coming to the 30th subunit or the 40th subunit, but it comes to what will be the P site. That is the only time. Okay. Then the next tRNA comes to the A site. The second tRNA comes to the A site. The dipeptide forms and it is moved to the P site. The first tRNA has now moved to the E site. The second tRNA has now moved to the P site along with the two peptides. Then the third tRNA again comes to the A site, and so on and so forth. It moves. Okay. So ribosomal RNA, they also have their secondary structure, and as I said, that the ribosomes can exist either as individual subunits or as two subunits together. And the Venki Ramakrishnan actually earned his Nobel Prize by deciphering the structure through electron microscopy. The mRNA, peptidyl tRNA bound ribosomes. Okay, like in the process of protein synthesis. So he could see where the deacyl tRNAs, the peptidyl tRNAs, and all of that. I am really not an expert on that, and I am not in a position to comment on it either. Structural biology is not something that I understand, but I know that Venki Ramakrishnan deciphered the structure, and it was a big deal. He was awarded the Nobel Prize. It was a very long-standing problem. And people got insight about how protein synthesis actually happens, right? So that's where we will end this lecture, and in the next lecture I will start with protein synthesis per se. Okay, thank you.